Hello, everyone. A very good evening to all of you. Welcome to the PhD Talks by NcodeX, episode 6. So before we move on to the program, first I would like to give a bit of introduction to who we are. So we, NcodeX, are a research institution where we develop cutting-edge research to bridge the gap between STEM academia and industry and to reach the sustainable development goals set by the UN. At NcodeX, we have major two arms. So one is the research and development sector, and the other is the STEM training. So at STEM training, we have many programs as STEM coaching, STEM mentoring, online courses, internships. So we have many more programs coming up as well. So for any inquiry or any information on what we do, you can always hop into our website, that is encodexpert.com, or else you can always reach us out through our social media platforms. And also you can drop an email to inquiries@encodexpert.com. So today, for the PhD Talks by NcodeX episode 6, we have a very talented speaker. So he is Mr. Lalinka Herath by name. He is a PhD student at University of Deakin, Australia, and University of Colombo, doing his Cottutelle PhD program. And also he has completed his master's in molecular genetics at Jeju National University, South Korea, and his bachelor's in molecular biology at University of Colombo. And also he is an Erasmus Plus PhD mobility scholar at University of Granada, Spain. He's also the former lead scientist at Molecular and Biology Human Genetics Unit at University of Colombo. So I will be handing over the mic to him in a very few minutes. So I'll give you a brief description of how the procedure of the program would be. So we have two phases. During the first phase, I will let the speaker to deliver his talk, and then during the second phase, he will be ready to answer any question which we can forward to him through our chat, or else you can also put your questions in our social media platforms in the comment section. So before taking too much of time, I will be handing over the mic to Mr. Lalinka Herath. Mr. Lalinka Herath, thank you very much for accepting our invitation to be a part of our program. And over to you, Mr. Lalinka.
Thank you very much, Ms. Lakshmi, for the lovely introduction. So today, actually, I'm going to talk on the topic that I have shared with you all before. So let me share my screen first. So is it there? Can you all see? Really, is it good?
Yes. Yes.
So, yeah. All right, then. So to start the talk, before starting the talk, what everyone wants to relax, and in Sri Lanka, this is a long weekend. Nobody wants to hear kind of big science, I guess. So I just want to start it like this. I think all of you have seen the movie Spider-Man. I think so. Yeah, I guess everybody is aware of the Spider-Man, the guy who can kind of shoot webs out of his hand, and still he's a human. And we all know who's this guy. And but have you ever thought, is this actually possible for someone to do, like a human to shoot webs out of his hand or something like that? And is that a superpower, or this can this happen in real life? Okay, then what about this guy? He's Wolverine in X-Men. So he has these long bones coming out of his hand. And if you watch the movie, you know that they talk about a gene called X gene, which is mutated. And due to this mutation, he's got some kind of superpower that he can use his bones, and he can actually, he's stronger. Nobody can kill him. Some kind of superpowers he has. So this guy is Wolverine. So these kind of people, can they actually exist in real life? So, and what about the movie Jurassic Park? So in that, you see there's part that there are dinosaurs in the park. So which the people have, which the scientists have brought them back to life extremely long ago. So can these kind of things be done really in science? Is it scientifically sound to bring long extinct creatures back to life? So is there a science, are there kind of hidden scientists, dark science work that they are doing this kind of research, that they're trying to bring them back and to give superpowers to human? Are there tools really to do these kind of things? So let's go into the topic and find out if there's really those kind of tools that exist for kind of manipulating organisms, living organisms, and then to give them superpowers, bring some kind of strange creatures back to life. So things like that. So jumping onto my topic, which I have given, which is gene editing in seed, that I'm going to talk about the environmental and also health applications of these gene editing techniques in an organism, which is a fish called zebrafish. So throughout this talk, you will find out if there is any tool that we can utilize to manipulate the genomes of living organisms. Because the gene editing is what you have to do if you want to kind of incorporate novel features into the organism, and then to study if there is any difference, anything that we can introduce into organisms that are not available, like new traits, new features. Topic. So I want you all to have a close look on the human body and ask the question, what are we are made of as the living organisms. We are the most, for me, we are the most complex animals, and then when it comes down to human, we are the most complex thing on the planet. But do we really know what are actually made of? I am different from you, and I am different from my friend. So what make me unique, and what really defined myself as an organism? Have you ever thought of these things? Like, there's no such complex thing like human or other animals on the planet. So I'm curious to find out what are the determining factors of our features, our traits, and what make me different from my brother and my parents. Why I'm look alike to my twin brother, things like that. So when you really have a close look on animals and humans, you understand that when you look at biologically, you understand we are made of the smallest living building blocks is there's something called cells in our body. So when you have a deep look, you see that we are made of billions of cells. And then when you really want to analyze further, what you really see is that inside the cells, you can actually see there's something called nucleus. So when you break open the nucleus and look at content inside the nucleus, you understand there's material inside the nucleus called chromosome. So these are like chemical materials. These are all chemicals. So if you want to study further, you can actually look deeper into chromosome and identify what's really inside the chromosomes. Inside the chromosomes, what we see is the material called DNA. So when you analyze the DNA, it is made of another chemical called nucleotides. And also when you study a segment of DNA together, like you understand that these DNA segments had certain functions in it that they determine certain traits, certain characteristics of animals. So what you see actually is this is where all the information about our behavior, our morphology, our characteristics are stored in. So this is a very important material in human body or any other animals or any other plant which is living organisms. So we have to study about DNA and what are the genes if you want to understand the behavior of animal and their unique features. So if you want to look at the structure of DNA, we call this is a double helix, which is kind of two cords twisted together and bounded certain bridges. And this structure is a stable structure that it can carry information in it about animals and plants and their behavior and also their other morphological characteristic. So to understand the concept of genes, you can actually think of the set of genes as a book that it has been written in a language with 4 letters in it. And if you read the entire book, you understand the entire story about a specific organism. So let's say that if you read one paragraph, you can tell about his eye color. If you tell another paragraph, you can actually tell about his or her hair color. Or if you read another paragraph, you can actually tell about these organisms, some unique feature characteristic like the way he or she speaks or responds to something. So that everything is stored in this book called Genome, which is made of genes. So it is written in 4 letters language. So in biology, from the definition, genes are the basic physical unit of inheritance. So these things can carry information from your parents to the offsprings. And this is where actually information like specific traits are stored and contained. And these are made, as I mentioned before, these are made of DNA, and it can actually carry instruction to some another chemical or another thing called protein that actually do the functions and determine our entire body characteristic or any other animal's characteristics. So basically on genes, that's where the information is stored. Proteins are what that the function, they are the one who coordinate the functions to actually determine the characteristic. So these genes can actually transfer from one generation to next generation. So when you celebrate having a being baby, what you actually celebrating is the transfer of one person's gene, half of one person's genome, along with another person's genome into a new living organism. So what they basically happen is it combines one's genetic materials with another one's genetic materials to give birth to another organism with the combination of gene pools. So how these genes actually work, actually function. So the information on the genes are stored on DNA, but they need to have a mechanism to transfer this information onto the functional sites. So there's one mechanism called central dogma of life, that genes can actually produce another material called mRNA and then produce proteins. So proteins are the things actually going to do the function in your body. So what happens actually when you have, if there's any mechanism to edit the information on gene? For an example, let's say there's a gene called gene A. So when you have a tool to edit these genes, we can actually change the genetic information on these genes. And then we can come up with the modified version of genes. So once the gene is modified, what happens is eventually the final product of protein is going to change. So the function this modified protein is going to get altered. Either it's not going to do the expected function, or it will do some strange function. So once the gene is edited, the protein's function is going to affect. So the expected or the real mechanism is not going to trigger in ordinary manner. So now I'm going to enter into the topic of gene editing. So gene editing is what scientists are actually studying these mechanisms to identify whether these gene altering mechanisms these are naturally exist in organisms. And then once they understand these mechanisms, gene editing is happening inside animals, inside plants, they realize that we can actually manipulate these techniques. Like what we do is we study these mechanisms from the living organisms, and then we come up with new tools that we can purposely, targetedly edit the genomes of animals and the plant. So actually there are tools that scientists have developed that we can targetedly edit the genomes of animals and plants. So this is the study area of gene editing begins. So let's say when it comes to gene editing, as in any other area, like let's say there's carpenter that he wants to build a chair or table, so he needs to have a certain set of tools to do this job. So exactly the same way, if you want to edit the genomes and genes, you need to have tools to do this job. So let's have a look on the tools that are available to edit the genomes of animals and plants. So when you look at the tools that are available to edit the genomes, there are 4 main types currently exist, which are known as meganucleases, zinc finger nuclide, TALEN, and CRISPR. So using these tools, actually scientists can cut and trace the component of genes. And also, let's say if you have, there's any gene component that you need to get rid of it. So you can actually cut it and remove it from the genome of an animal. And also at the same time, if you want to incorporate a new kind of foreign DNA fragment or gene into a genome of another organism, you can utilize these tools. You can use pull one of these tools and then cut and paste the gene of interest or gene component of interest into the genome of a specific organism. So in this way, we can actually come up with new traits, new attributes, and organisms with new functions, which are known as transgenic organisms, because they are genetically modified in a way they are genetically improved. So we can actually utilize these organisms for several functions, several applications. So if I show you quickly what is the most popular gene editing tool at the moment looks like. So it has a DNA, it's a kind of molecular complex that it can come and integrate with the DNA and then cut it into pieces and then repair it with some mechanism. So which has something called guide RNA and Cas9 protein. So this tool is known as Crispr gene editing tool, and it's becoming quite popular among scientists who want to edit the genome of animals and plants to the date. So I am not going to explain further on the techniques because these are kind of that I can say complex, but yeah. So I'm not going to elaborate on those techniques further, but I think that you might find it interesting to know what are the applications look like once the gene edited organisms are modified with these tools. So the application can be categorized into two main groups, which are plant science gene edited plants and gene edited animals. So if I, as example, if I look at a couple of examples, so you all know the rice plant. So rice plant has been modified with certain gene editing tools for it to become pesticide resistant and to give more crop. And also there are modifications have been integrated using these type of tool for them to become more tolerant to environmental conditions and such things. So these are so when farming, when you actually farming, when it comes to rice farming, it's these advantages when they are genetically improved. Actually, we don't say they are genetically modified, they are genetically improved because these are improvements. These can happen naturally as well in the organism. We what as scientists what we do is we targetedly improve their qualities in the organisms. So some of you may have seen the tomatoes that have been in the market that have been genetically improved to change the size of it, and also when it's transporting they can damage. So these kind of tomato can be modified with gene editing tools for them to become bigger and easy to transport vegetables. So those are two examples for gene editing in plants, and I would like to show you some gene editing examples in animals as well. So if you look at this cat, so it has been modified genetically to glow in dark, and as you can see, it's glowing in fluorescent green color. But this has not been modified to take to a pub or club. The purpose for it to be modified for scientists to study the mechanisms actually happening behind the HIV resistivity in human and animal. So what they have really done is they have coupled the genes related to HIV resistant pathways with fluorescent genes. So once HIV is there inside the cat is going to glow. So scientists can use this as an indicator to study the presence of HIV virus inside the cat. So this is another interesting application that scientists have developed a genetically modified mosquito species using gene editing tools like CRISPR gene editing tool, that this can actually kill the dengue virus when it is entered into the mosquito's body. So this is actually a tool we can actually apply to eradicate dengue in future. And when it comes to human related application, one major application of gene editing is that gene therapy, where we can utilize these tools to correct one or two mistakes in several disease conditions of human, like brain diseases. We can utilize gene editing tool to correct brain diseases, and also to improve the eyesight of some patients, and also if there is any defects with livers, there are actually specific gene editing techniques that we can apply and correct these problems in human. So this is one of the major application of gene editing tools in human. So gene editing in zebrafish. So this is the topic what I want to explain to you, that what I have been doing in my PhD study as well. So zebrafish is one of the organism which is a small fish that you can find in fresh water, mainly in South Asian region. So this is a small fish that you can also genetically manipulated genome in order to use it for a few applications. So what are the features of this fish? So the scientific name of the fish is Danio. So this is a small freshwater fish, and it is 4 centimeters long when it is fully grown, and very easy to look after. So what are the applications of zebrafish when it comes to gene editing? So the zebrafish genomes has been very well annotated by scientists because it's a small fish. We know the entire genome, which is the full pool of genes in this fish. So we can actually use this fish to study several things by changing the gene functions of these inside this fish. If I have a quick look on the applications, like general applications of genetically improved zebrafish, so it can be used in a couple of environmental application that we can use these genetically modified zebrafish line to detect the toxicity of certain toxins in contaminated water sites and others, and several pollutant detections. And also we can, at the same time, we can use zebrafish, the genetically improved zebrafish, as a disease model for certain human diseases. Let's say we can actually study human cancers by integrating the same disease conditions in a zebrafish, by modeling a zebrafish, by editing their genes, which mimics the conditions of a certain cancer complications. So in my PhD, what I actually did was I utilized zebrafish for some interesting environmental application. I genetically engineered the zebrafish in order to detect heavy metals in contaminated water samples. So what I actually did was I identified a gene which is responsible for heavy metal detection in organisms, and then I coupled that gene with a fluorescent protein producing DNA coding sequence, and then I integrated this entire mechanism into the genome of a zebrafish. So now this novel zebrafish line is genetically modified. So this can actually function as a biosensor to detect heavy metals in contaminated waters. So what happens is when heavy metals is available in a contaminated water sample, the wild type zebrafish is going to glow in red color so that we know the water has been contaminated with heavy metals. So coming on to the last slide, what I want to discuss is the responsible use of gene editing. Actually, these tools are very powerful tools that we can utilize to change the attributes of an animal. So we cannot, being in the scientific community, we cannot actually, like we talk, we cannot actually haphazardly use these tools in gene editing of animals or plants. So what happens is if you utilize this tool irresponsibly, you can actually end up imbalancing the nature's mechanism. So we have to be very careful when we edit the genome of an animal or a plant. So because we are actually changing the natural mechanism of the nature. So to regulate these kind of scientific project, we have to think of three main aspects, which is bioethics and the work safety and also gene technology regulation. Bioethics is where that you have to think of how you are going to ethically use the organism. Let's say you're going to edit the genome of a fish or genome of a mouse or another organism. So you have to ethically do this. You are not going to go into the lab and edit thousands of rats, thousands of fish in a row. You have to do the math, what number of fish, what number of animal you are going to need to come up with the object to cover the objective of your research, and do the justification and use only the amount that you need. And you have to make sure that you are not going to hurt the animal. So we have to consider all these things. And also when it comes to work safety, we have to care about the animal and also the humans working in the lab. And gene technology regulation, which is the most important part. So if we are going to do a gene editing, if we are going to edit the genome of any animal or plant in the lab, we need to go through a process to get the approval. Like in the countries like Australia, they have their government gene regulatory body, which is OGTR, and in the countries like Sri Lanka, we have several ethical committees. So we have to get the approval of these committees and the bodies before performing any gene editing experiment. If not, what happens is people can do dangerous things, like they can actually produce bioweapons, like by editing the viral genomes. So these kind of things need to be regulated. So this is the comes to the end of my presentation. And so if you want to learn more, if you are really interested, you can watch this TV series called Biohackers, that people unethically edit genes and what are the consequences. So it's a Netflix series. You can actually pop in and watch it. So thank you, everyone. So that's the end of presentation.
Thank you,
I
Mr.
want to thank,
Lalinka.
give me a minute to thank my supervisors. My supervisors are Professor Anil Dasanaka, Professor Nalinka Silva, Professor Ongni, Professor Nilmini, and also Professor Luis and Dr. Iran. So they are the kind of supervisory panel who are like the experts in these areas in the world. Thank you.
Thank you very much, Lalinka. So that was a wonderful talk. And so we love the way that you had organized your speech from the very basics to the very complicated ones. Yeah, so that anybody, outside the STEM disciplines can also understand what's really out there in the world of genetics. Thing. So moving on to the questions, we have a question. Now you said we have to be very mindful when we select traits when introducing into genes, right? So are there any practices that as scientists that they follow to ensure that the unnecessary traits are not introduced into gene editing, like to make sure that they are not introduced?
Yeah, that's a very good question. Actually, yeah, to identify what genes to edit or alter. So you don't really do it first and see. By based on the result, you are not going to decide, oh, I edited this and I produced something else. Now I'm going to edit this and see, is it going to be something else? So that's not the way to go. What to do is there are uh, some other before that preliminary studies that you need to do before picking up the genes for editing. So if I say there are studies called Jiva studies that you can actually uh, scan the genome of an animal. So by scanning, you can actually, if, as an example, when it comes to my study, what I did was I developed a zebrafish that can actually detect heavy metals in contaminated water samples, right? So the gene I utilized was MP2 gene, so methylmethionine resistivity gene. So this is one of the genes in the pathway of heavy metal regulatory homeostasis pathways gene. So to identify these genes, what we first do is we will go dig into the literature if there's any information that we can retrieve to identify very well annotated heavy metal sensitive gene. If there's no such gene, what we are going to do is we are going to inject some animal like rats or other model organism with heavy metals, and then we express their genome to see if there is any novel genes getting upregulated, which is corresponding to the required trait. So once you know that there are, let's say 5 genes are getting upregulated. So those are the genes you are going to pick and then going to utilize in the gene uh, editing experiments. So these are kind of preliminary lab work that you have to do before entering, like touching the gene editing parts of the real organisms. So that's a good question.
Thank you, Lalinka. And also now this model that you talked about, so is it already in use? The zebrafish
Yeah,
and the
it,
mechanism?
it's functional. It's functional in the lab. So we have actually confirmed the mechanism and we have calibrated it for heavy metal detections, but we haven't actually, it's at the laboratory scale. We haven't taken it down to the site, but our
So
plan is,
what's pending is to take it out to the next level.
yeah.
Is it?
So yeah, like now this is a living organism. So your point is that we need to couple it with some kind of STEM application. We need to come up with a digital device which is portable that we can couple with the fish, and then kind of capture the signal coming out of fish. So these kind of things are in discussion with the other groups like engineers in the other universities, and there are scientists who are working on photonics. So we are actually discussing how we can utilize this outside the lab. For the moment, what we are doing is we are capturing the images of these fish from the fluorescent microscope, and then analyze these fluorescent images to identify the signals coming out of fish. So our ultimate target is to come up with a digital device that we can couple it with the fish and then take it to kind of on-site detection application. So when it comes to, yeah, it is very important that we come up with STEM approaches, science, technology, engineering, and maths. So we have to actually communicate. It's not just do biological, we are biologists. It's not just we are doing play around the genes and come up with organism, throw it away. Then we are responsible about this because we are bringing a new organisms to the world. So we are responsible about their fate. So if we are not going to utilize them in the field of the betterness of the humanity or something, no point of bringing the organism. So these STEM applications are very important. Actually, that's why I'm not going to throw the fish away after my PhD, that I want to push it further. Yeah.
Yes, Lalinka. So that already answered our next question, which is on interdisciplinarity of your research. So yeah, so it's a good approach that we can merge all the scientists from science, technology, engineering, maths into one research and make it a more impactable research, right? How can innovation be brought to your research? You were talking about a detector or sensor kind of a thing. So how can you bring innovation into research and maybe at this stage or over the overall project?
Yes, innovations in the sense actually, this is actually when you are designing a gene circuit to edit, there's something called gene circuit in gene editing that you are designing, and it is a novel segment of DNA that we are incorporating into a new animal. So once they are modified, so this animal is a novel organism. Uh, so it is going to be a new kind of inventions, but right now we cannot, in some continents of the world, we cannot actually patent the living organisms. So what we do is we can actually patent the novel gene circuit. We can integrate it into the genome of these animals. So it's an innovation at that stage. And also we can actually couple it with kind of another device, because when it comes to the comes to real world applications. So for as an example, one of the chapters from my PhD studies is that I designed a bacteria which is genetically modified. Since it's a small organism, so we could actually design another device which is a portable device that we can couple it with the bacteria in it, and then take it to the site. We can plug it to the computer and then detect the again for detecting heavy metals in contaminated water. So these devices that we have already developed, but for fish it's a bit tricky because we have to think of certain facts. So anyways, so those are the things that we can actually do. We have to communicate with the other scientists from other disciplines. Like it's very important for biologists to work closely with electrical engineers and yeah, like mathematicians, because the device I developed, that I had to develop an electronic device, and also then I had to couple it with a fish, and then also to kind of narrow down the signal's specificity, I also had to develop kind of come up with new mathematical equation. As a biologist, kind of a nightmare for me, but what I did was I went to the mathematical science department and then I talked to people in the department. So it was like just piece of cake for them to come up with the, but so this is how it should work in science. We should work with other people from other discipline as well.
Yeah, that's a really interesting research. So from your research, what can you give out to the people as the message with regard to your research? From your findings, maybe?
Yeah, fine. This actually this is a translational science research, which there's not much pure science in it, but this is a very important application that I have come up with. So what I expect is I'm going to pattern the technologies I have developed through the chapters of my PhD to detect which are novel heavy metal detection mechanism, and then to come up with new devices and that can actually people can use in future for their day-to-day life to make sure the water quality water they consume is safe and also pure for them to consume. So so that's the impact I see from my research, what I have done in my PhD.
Okay, thank you, Lalinka. And there's another question coming out. So what is your opinion on how to use this zebrafish model to determine water purity in rural communities like Nigeria? Like how you can use this model to determine water purity in rural communities?
Yeah, that's a good question actually. For the moment, what we have is we are not using the adult fish right now for heavy metal detection applications in the lab. We are producing the larvae like 72 hours post fertilization old zebrafish, like the small like larvae zebrafish for the detection application, that we can actually utilize them only in the laboratory. So we have actually developed a laboratory assay right now. We can incubate these zebrafish embryos, which are the babies of the fish, in small plates, and then treat them with heavy metals, and then check their signals under microscope. To utilize the technology in the rural areas, what we have to do, we should come up with a device which is affordable for the community. So that's what we have been working. Right now, we are kind of prototyping a different kind of device in the lab that I cannot actually explain because these are in the experimental stages. So we are actually kind of working on those aspects. So our ultimate objective is to take it to, it is affordable and accessible to everybody, even the rural community, not even in Nigeria, South Asia, and it's gonna be a day to day, like day to day device in your household.
Yeah, so how would these outcomes be helpful in the future sustainability?
Yeah, so water quality, water. So this is a water quality assessment project. So uh, ensuring the quality of the water is very important because it's the basis of life. So there's no argument that we need to come up with as much as tool to make sure the water we have on the planet is safe and pure, because we can't actually, we have the water on the planet that is recycling, so we have to make sure we are actually looking after water we have on the planet for future generations as well. So it's no harm come up with as much as tools to uh, make sure water quality is good, and so certainly it's a sustainability related project.
Yeah, thank you very much, Lalinka. And so at NcodeX, we have quite a number of young researchers who are just entering their research careers. So what would be your advice to them, irrespective of the STEM discipline? So they could be from science, tech, engineering, maths, any discipline. So what would be your advice with your experience and the knowledge?
Yeah, like it's the advice in the sense what we can't actually, I mean, when it comes to science, different, my journey as a PhD student is different from another student. So experiences for me is entirely different what Ruvini encounters in the lab, because her project is entirely different. So mine is entirely different. So what the message is, what I have for people who are like students, like young scientists who are starting their PhDs, maybe just about to start their postgraduate, my message is, do not ever compare your project or your what you've been doing. So what I mean is, don't stick to your timeline, because your project is different from another person's project. So don't worry about what happenings, what going on. The other person is getting a lot of results, so I must get, because it's different, because PhD is something like that. Everyone is going through a different journey, their own journey. So do not ever compare yourself with anybody else. Just follow your passion and your path. That's the message.
Yeah, that's a very good message that we all can take home. Yeah. So I hope there are no other questions from Facebook. Are there, Ruvini? No, right? Yes. So then we can wrap it up. Ruvini will be posting the link to the feedback form. So she'll be posting it on Facebook as well. So if there's anybody who would like to give us feedback, because your feedback will be very valuable to us in improving our future sessions. And at the same time, she'll be sharing the link to the form of registration for PhD talks, where if there's anybody who is interested in delivering a PhD talk, can go through the link and fill it up so that we are aware of your availability and we can get back to you as soon as possible. So today, I thank Mr. Lalinka Herath for joining us with the program in short notice. Thank you very much for accepting our invitation with no hesitation at all. We wish you a very good journey and thank you very much.
Thanks.
Stay safe.